Okay, thank you, Christian. This is the first time I'm formally meeting Christian, but I think I've known you for a while now, because a lot of the biochars that are sourced in this project are from the UK BRC. So I've been in communication with Christian for some time, and I've been attending these seminars for a while, on and off for a few years. So thanks for organizing them. They are an amazing opportunity to learn about biochars and to also, for my first time, present my work. So thank you all here for your time and attention. So personally and professionally, I'm motivated by technologies and systems that are designed to serve the most marginalized. And sometimes I use the United Nations Sustainable Development Goals to assess these technologies. Creating and using biochar is one such technology. Specifically, if we think in the context of source separating toilets, where we gather urine and feces separately, the feces can be converted into biochar, or biochar can be created using any other feedstock that we might know of. That biochar can then be used to recover nutrients from urine, specifically like nitrogen, to create a nitrogen-enriched soil amendment, which can be applied to agricultural soils. And this process can create the biomass, which can eventually be turned back into biochar, creating a more connected or circular bionutrient ecosystem. One of the limitations to the system that I chose to focus on stems from the knowledge that biochar and thus biochar systems and responses are highly variable. Specifically, I'm referring to biochar sorption of nitrogen, like the ammonia found in hydrolyzed human urine. This variability is because biochars can be made from many different feedstocks, including sewage sludge, like is done at UKBRC, from agricultural wastes and so on. It could be made using different pyrolysis methods and parameters. It could be modified physically, chemically, biologically, pre and post pyrolysis, and then characterized in many different ways, including at the elemental or at the surface level. So it's almost no wonder that we see large variability in biochar's sorption of nitrogen. What we see from the literature is that there is wide variability, in biochar's reported nitrogen sorption capacity and conflicting evidence regarding the characteristics and mechanisms that drive nitrogen sorption. We see sorption capacities reported here on the y-axis in milligrams of ammonium per gram of biochar that span orders of magnitude when looking across different biochar feedstocks. Over the years, we've been converging on the concentration and protonation state of biochar's functional groups, exerting strong influence over its sorption of nitrogen. These functional groups can be measured in different ways, some shown here on the left, including the more commonly used BAME titration, which is an indirect titration method. We've seen some more sophisticated subclassifications of functional groups by the likes of Rivka Fidel and others, as well as updates to existing methods like the modification to the measurement for protocol for measuring cation exchange capacity. And these are just a few examples. So we do have a number of studies that are able to relate these functional groups, however they are measured, to nitrogen sorption. However, many of these studies look at biochar sorption of nitrogen in simple ammonium solutions. Urine, however, is more complex, partly because it exists at a pH where ammonia and ammonium, or total ammonium nitrogen, the acid-base pair, are present, and because it has a high buffering capacity, mainly due to this total ammonium nitrogen pair again, and it has a high ionic strength, again, primarily due to the total ammonium nitrogen pair. These are factors which would interact with biochar chemistry in ways that are distinguishable in ways that distinguish the total ammonium nitrogen sorption behavior in urine from the simpler ammonium solutions that are more commonly studied. Now, there are some studies that do look into biochar absorption of nitrogen from urine specifically, but these studies are a bit limited in that they're unable to make broader generalizations of their outcomes because they often only study one type of biochar, which brings us back to the earlier point of biochar systems and responses being highly variable. So given given this diversity of biochar, the wide variability in sorption capacity, prevailing evidence of the role of functional groups, and the limited studies on urine specifically, I sought out to better understand how the concentration of biochar's functional groups influences its sorption of total ammonium nitrogen from hydrolyzed human urine. I chose to focus on three key aspects that would address the variability we see in existing studies, as well as make the results more representative of full-strength urine. This includes looking at what I consider a diverse suite of biochars, looking at multiple ways of characterizing biochar functional group concentration, and studying sorption dynamics under conditions representative of hydrolyzed urine, which I'll get into a bit more. I focus on these aspects to be able to assess the effects of functional group concentration, regardless of all these factors that influence biochar characteristics. In total, I look at 4 different biochar feedstocks. And so I'll take the time again to thank the UK Biochar Research Centre, who provided 3 of the 4 feedstocks, without which this project as is would not have been possible. So 4 different biochar feedstocks, 2 different pyrolysis temperatures, and 2 different pyrolysis methods. I oxidized the biochars with varying concentrations of hydrogen peroxide to modify the functional group concentration in a controlled manner. And in the end, create a set of 19 different biochars with a range of functional group concentrations. I analyzed the biochars, the concentration of functional group of the biochars based on the bulk oxygen to carbon ratio, or O to C, the surface O to C, and the proton density, or the concentration of proton active functional groups. This measure of proton density is based on direct titration of biochars as depicted in this graphic of our system, where briefly we equilibrate the biochars in a dilute salt solution to be able to measure a stable pH. We sparge the biochars with nitrogen with N2 gas for 2 hours prior and throughout the titration to control or to rather ensure there's no contamination of proton active dissolved gases like the carbonate system, which is a decent buffering system in the pH range over which we're titrating. And we titrate with either acid, strong acid, so or strong base down to pH 3 or up to pH 11, respectively. And what we see from our titrations when we look at the proton density or net apparent proton charge on the y-axis in millimole charge per gram biochar and pH on the x-axis, and the bulk oxygen to carbon ratio as a color gradient from orange, yellow, low in orange and high in blue, to systematically distinguish between the different biochars, is that majority of the functional groups had pKa's and thus buffer capacities in the pH 3 to 7 range. And this basically means that the majority of the functional groups would be deprotonated in urine's pH, which is roughly 9. We also see from looking at the neutralization plot, this time with the change in pH per mole of titrant added on the y-axis, is that for more oxidized biochars, again, those in the darker blue hues, we see a continuous distribution of proton affinities across the pH range. And that's indicated by the increase in the neutralization point. So this is a neutralization point, this is a neutralization point. So we see an increase in the neutralization points for the more oxidized biochars. And this suggests an increase in the diversity of discernible proton active functional groups. What this means is that direct titration may be able to sufficiently both quantify the concentration of functional groups, as well as discern the diversity of functional groups across biochars. To be able to relate proton density as well as other measures of functional group to total ammonium nitrogen sorption capacity, we generated sorption isotherms in batch experiments. First, we did this by diluting urine, kind of depicted by the change in color here, and equilibrating it with the 19 different biochars. And from these experiments, when we look at the sorption density on the y-axis in millimole of total ammonium nitrogen per gram of biochar, as a function of the total ammonium of the TAN concentration, which we depict as, or we represent, excuse me, on the x-axis as equilibrium activity. And we choose equilibrium activity over concentration because activity takes into account ionic strength, buffering capacity, and pH of the solution in equilibrium with the biochar. So when we look at sorption density versus TAN concentration for equilibrium activity, we see that the sorption density across the biochars starts to plateau as we approach the concentration of full-strength urine. This behavior is often characterized as the Langmuir-like isotherm. We also looked at sorption isotherms by changing the dose of biochar and equilibrating it with full-strength urine. And what we see here is an increasing sorption density as we approach the concentration of full-strength urine, which is a contrast to the Langmuir-like behavior which we saw in the previous plot. While this behavior is intriguing, the cause of this high sorption density region, higher than what we've mostly seen reported, after exploring some mechanisms and reanalyzing some published data which showed similar behavior, remains unknown. So we're still unsure of what's causing this high sorption behavior. And it warrants further investigation because verifying this behavior and identifying the underlying mechanisms driving the behavior could have important implications for optimizing biochars for TAN recovery from urine. The main takeaway from this aspect of the study is that we can say that across the 19 biochars studied, biochars are able to recover TAN from hydrolyzed urine. And to be a bit more quantitative about that, we correlate these sorption densities across the multiple ways which we characterized the biochars, and find strong correlations between functional group measure and sorption density. These correlations hold true whether we are looking at the sorption densities on the y-axis in millimole TAN per gram biochar as a function of either the bulk oxygen to carbon ratio, surface oxygen to carbon ratio, or the proton density, as well as whether we are looking at sorption densities from diluting the urine or sorption densities from changing the mass of biochars in equilibrium with full-strength urine. And what we're able to say from all of this is that if we have a biochar with a bulk O to C ratio between 0.13 and 0.31, a surface O to C ratio between 0.15 and 0.35, and a proton density between 0.01 and 0.25 millimole charge per gram biochar, is that you can expect to see absorption density within this range. So the main takeaway from this aspect of the study is that these three independent measures of functional group concentrations directly correlate with total ammonium nitrogen sorption across biochars. So to reiterate the main takeaways across the entire study, I summarily say that direct titration, distinguished functional group concentration, and diversity. And more broadly, one can use acid-base titration of biochars or any of the three characterization methods presented here, maybe based on what is accessible or available to identify optimal or to rule out suboptimal biochars and thereby simplify the biochar uh, selection. Yeah, the biochar selection for total, specifically for the case, the instance of total ammonium nitrogen recovery from urine. And thank you. I want to acknowledge my funding sources. And then I also want to end with kind of saying there's a small group of folk and I through what we call the Circular Bionutrient Economy Network. We're based in Kenya and we're hosting a joint conference with the International Biochar Initiative in June of this year. And I placed a link here for more information. And I'm also currently based at the World Agroforestry Center or Center for World Agroforestry Center, doing some predictive modeling of biochar characteristics using infrared spectroscopy, basically trying to see to what extent we can characterize biochar property spectroscopy. And I hope to present that work at a later seminar. And I'm also submitting my dissertation at the end of this academic year and putting my next steps in place. So if any of this sounds intriguing or interesting enough to discuss further in future research endeavors, please reach out. And thanks again for your time.
Thank you very much, Silva. I think it's very interesting. If there are any questions, please write them into the chat. My first question would have been so which biochar was the one that worked best so far?
That's interesting that I don't look at that. I would say, yeah, it was between, and you are very familiar with these, but it was between the softwood pellet and the rapeseed straw. But that's the thing. Yeah, I was trying to kind of get away, and that's what I am trying to claim here is, well, there's a big delay, is trying to get away from, because you can say it's either which feedstock works best, which I think is kind of the question you're asking. And that's, oh, yeah. Yeah, that's what I'm trying to kind of navigate through is to, can you hear me okay over the noise?
Yeah.
Okay, that's what I'm trying to get past is that I don't know if looking at feedstock alone, is as informative. So my goal was to try to say, or my initial goal and how I approached this research endeavor was to try to say, is there one parameter that we can look at? And for me, that was the concentration of functional groups, and you can either measure it using O to C or proton density. So I would rather answer that question in saying that the biochar that is able to absorb more, generally the biochars that are able to absorb more nitrogen from urine are the biochars with the higher functional group concentration. And in this instance, that happened to be the softwood pellet and the rapeseed straw that were oxidized. I don't remember the exact concentration they were oxidized to, but I think it was the most oxidized biochars because generally you increase oxidant concentration, you get more functional groups. So the biochars, in summary, the biochars with the higher functional group concentration is how I would answer that.
And do you have plans to use oxidized biochars that are produced in so I'm thinking about open flame cap kiln biochars that are always
Yeah.
somehow oxidized to some degree.
Yeah, so that was another sort of like a, a research decision. And I chose to start off with the UKBRC biochars because I consider them the like standard biochars. And although I did modify them and we can't now relate the results to the unmodified biochars directly, um, I had to choose between sourcing biochars that naturally have a high or like a relatively high O to C ratio. And I think one iteration of this project would have been no oxidation, like no chemical modification. And I was just able to source biochars that had a diverse enough range of O to C of functional group concentration, because that's my x-axis. And I think that was challenging. That was a little bit more challenging. And then I would have I think I would have had to talk more, a little bit more about controlling for, I guess no, I wouldn't really have to talk about controlling for functional groups, but it was slightly easier to source a few feedstocks and then modify the functional groups than to reliably source the like if I needed to directly source 19 different biochars with a diverse enough range of functional groups. And not only that, it wasn't one of the limits of my results that I would speak to is that I don't think that our functional group concentration range is that diverse. So like for if we look at bulk otocetes between 0.15 and zero point 0.3, and that is a decently wide range. But like when I start to look at my correlations, the plot is a bit ideally, if I was able to study a wider range of functional groups, but I do, if I'm looking at some review papers, this range is relatively representative of what is produced, and I think you can tell me otherwise if that's the
Uh,
case. But yeah, so it's not, I don't know, do you know of any biochars that exist within I think that is what you're kind of getting at. The biochars that exist with the O to C between, let's say 0.25 and higher, 0.25 to 0.35. Like what would be good source of those high oxygenated biochars?
I mean, we will have to check. I was just wondering if a different
Yeah.
production method would naturally increase the O2C ratio, and I'm pretty sure it does. But
Mm,
but yeah, I mean, I can have a look. I think we have to go through the questions because you got some in the chat in the meantime. Um,
I did.
so John Hoffmeyer was asking, please speculate about the applicability of this research in underserved and less sophisticated societies using unmodified data. So that, yeah,
Yes.
goes back to the last one.
Yeah, thanks for your question, John. And that was that is the goal of my of the projects that I approach. I try to approach them to be applicable in underserved and less sophisticated societies. Um, and in my hope is that being able to look across. So one other thing that let me try another way to approach this. One other thing that we looked at was the just FTIR spectra of these biochars, because there is that question of are these findings only applicable to chemically modified biochars, and specifically biochars modified with peroxide, because you can oxidize biochars with acid, or there's other ways of oxidizing biochars. So are these findings still applicable? And um, I mean, I don't, my research doesn't speak to that directly, but it is my hope that at least looking across different biochars, like that's the different feedstocks, the different temperatures, and the different pyrolysis methods, which are feedstock and pyrolysis temperature are generally the one of the two main factors that influence biochar physical chemical characteristics, specifically as it relates to oxygen. So it's my hope that looking across these different biochars and seeing this trend, if we take it to either biochars that naturally have the same or similar range of O to C ratios, or we take it to biochars that were oxidized using other methods, that the trend would still hold true. But I think that would to be able to say that with a level of certainty, we would need someone to source biochars with a diverse enough range, study, and then do the sorption of nitrogen. But I was mentioning the IR spectra is and just a bit of referencing the literature is there aren't, as it pertains to functional groups and the type of functional group and the functional group concentration. I don't know if chemically modifying the biochars results in a biochar that is functionally that, and I say this with a lot of caution, is functionally that different from a biochar that is modified in another way, either by how the pyrolysis process itself. But yeah, I think we would need someone to actually do that study across unmodified biochars and see if this is still applicable. But for me, now the goal in less sophisticated societies, as you put it, John, is to source biochars with a higher O to C ratio. And I hope that titration is a method that's maybe slightly more applicable. And I hope I'm also getting to Julia's questions with which analytical methods were used. This bulk ODC is just elemental analysis, which is combustion, via combustion, excuse me. And then surface ODC is X-ray photoelectron spectroscopy. And this is corrected for ash. So it's assuming it accounts for the ash content. The titration, kind of also still talking to John's thing, I'm hoping that titration is a little bit more accessible. That's a personal statement and not so much a research statement, but I am hoping that acid titration is, I would consider a less sophisticated method than elemental analysis and XPS. So hopefully it's a way that can be used to identify higher O to C ratio biochars. And then I think lastly, for the applicability of this in underserved societies is that you at least know, like, instead of having to measure all these other parameters, because that's the thing, biochar does have a lot of physical chemical characteristics. Instead of having to kind of like, which parameter are we looking at to know if this is good for this purpose, I'm hoping that this research can say that this is the functional group concentration, in whichever way you can measure it, is kind of the key parameter that you should be looking at. And ideally, you are able to access one of these methods to be able to measure it. But then what I did to kind of take this research a bit further to make it more applicable is that that predictive modeling project, because if we can use IR spectra to be able to predict either to be able to predict some metric that relates to functional group concentration, then that makes this a little bit more accessible. I can't remember the prices that are quoted at the Center for Analyzing Biochar, but it is generally much cheaper, significantly much cheaper than being able to get the O to C ratio by wet chemistry analysis. So a lot of different factors, but I hope I got to speak to both of those questions.
No, that's great. I think we are. Maybe you can keep stay in the chat for a bit, because you have, I think, 3 questions more. But we are perfectly on time now, so I think we have to jump to Alex Brody's presentation. So thank you again, Soliver. Very nice presentation.